The locust body is divided into three regions: the head, the thorax, and the abdomen. Like all arthropods, the body is segmented and covered with a chitinous exoskeleton composed of rigid and articulated plates, the sclerites. Each segment is formed of a dorsal sclerite, the tergite, lateral sclerites, the pleurites, and a ventral sclerite, the sternite. Flexible membranes link these plates to each other. The presence of 3 pairs of legs in the thorax is a feature of insects, also called hexapods. The head bears sensory organs and mouthparts. 3 zones are visible in the median plane of the head: the vertex, the frons, and the clypeus. On each side there are the cheeks, or gennae. In the lower part of the head are the mouthparts of the masticatory system. Sensory organs include 2 compound eyes, 3 ocelli, and a pair of antennae. Compound eyes are located on the sides of the head. They are formed by a large number of photoreceptor units, the ommatidia, allowing insects to have a wide field of view without moving the head. The locust has 3 ocelli, 2 lateral and 1 median, that are sensitive to light variation. The antennae are short and articulated. The first segment, the scape, articulates with the head. The antenna continues with a short second segment, the pedicel, and with a series of segments forming the flagellum. The thorax is divided into 3 segments, or metamers, that are called prothorax, mesothorax, and metathorax. A large shield-like tergite, the pronotum, covers the prothorax and a part of the following segments. The sternites of the mesothorax, metathorax, and the first abdominal segment fuse into a ventral plate, the sternum. The 2 pairs of respiratory spiracles of the thorax are visible anteriorly to the meso- and metathoracic pleurites. Each segment of the thorax bears a pair of articulated appendices, or walking legs. The legs, built on the same organization plan, are of different sizes. The segments are clearly visible on the hind legs that are the most developed and permit the animal to jump. From the base to the end of the leg, there are the coxa, the trochanter, the femur, the tibia, the tarsus, and the pretarsus. The tarsus is composed of 3 segments, having membranous pads. The pretarsus has 2 claws and a median pad, the aerolium. Each of the 2 posterior segments of the thorax bears a pair of wings. The forewings are sclerotized and are called tegmina or elytra. The hind wings are larger and membranous. During flight, the wings are held at right angles to the body. The tegmina are slightly raised so that the hind wings can flap. At rest, the tegmina cover and protect the folded hindwings. The structure of the wings is strengthened by thickened, tube-like veins. The abdomen is divided into 11 segments and presents a sexual dimorphism at the end. The first 8 segments are identical in both sexes. Each segment is composed of a tergite and a sternite. Pleurites are absent. The first abdominal segment bears the tympanal organ, with auditory function on each side of the tergite. The first abdominal spiracle is located anterior to this organ. Each of the following 7 tergites also bears a pair of spiracles. Tergites 9 and 10 are narrow and are partially fused. The last tergite forms a triangular dorsal plate above the anus, the epiproct. The 2 small plates on each side of the epiproct, the paraprocts, represent the remains of the 11th sternite. 2 elongated appendages, the cerci, are present on the flanks of the abdomen next to the paraprocts. Males and females display a sexual dimorphism on the last abdominal sternites. In males, the 9th sternite is divided into 2 parts. The distal part forms a subgenital plate that protects the external genitalia. In females, the last visible sternite is the 8th one. The end of the abdomen bears the ovipositor, the organ used for egg-laying. The ovipositor consists of 3 paired elements called dorsal, internal, and ventral valvulae. These structures are used to penetrate into the soil and deposit eggs. Flexible membranes are present between the abdominal segments, allowing an important elongation of the abdomen for the burying of eggs. The locust has a herbivore diet. The grinder mouthparts are pointed downward in a hypognathous disposition. This head region is comprised of several articulated pieces adapted for biting and chewing plants. The first mouthpart is the labrum, or upper lip. Behind the labrum are visible 2 triangular-shaped powerful pieces, the mandibles. Behind the mandibles is a pair of maxillae, each of which bears a 5-segmented sensory palpus. The masticatory system ends with a single piece called the labium, or lower lip, that bears a pair of palpi, each with 3 segments. These mouthparts will be dissected, then placed in a drop of water on a glass slide to be observed with a binocular microscope. The labrum is cut on its base. And then placed on a glass slide. The mandibles are moved aside and pulled out with a pair of fine forceps. The movements of the mandibles are ensured by powerful abductor muscles attached to a chitinous apodeme. Once the mandibles are placed on the slide, remove these muscles to observe the apodemes. In the middle of the mouthparts, one finds a tongue-like structure fitted with spines, the hypopharynx. Then, take out the maxillae by pinching on their base. The apodemes of the maxillae are also cleaned from muscles. Finally, after cutting the neck's synovial membrane, take out the labium and place it on the slide. The labrum has a concave shape and pushes the food back between the other mouthparts. On top of it is the clypeus. The mandibles are the most developed mouthparts. They have a serrated part to cut the plant and a molar part to grind food. The maxillae help to shred the plants. In the basal part of the maxillae are the cardo and the stipes. On the stipes are articulated the lacinia, the gallia, and the maxillary palpi, which are touch and sensory organs made up of 5 segments. The labium is an odd piece. Its basal part consists of the postmentum. The distal part, the prementum, bears the glossum, paraglossum, and 2 3-segmented palpi. The locust must be opened to observe its internal anatomy. First, cut the elytra and the hindwings at their base. Cut the integument with thin scissors at the level of the epiproct. Make a longitudinal cut on the mediodorsal wall. Extending the cut up to the head. Next, move aside the integument with pins to fix the animal in place. The pins are placed starting from the thoracic region down to the abdominal section that must be gently stretched. The rest of the dissection is made in water. In the general cavity of the body, the hemocoel, one can find the digestive tract, the gonads, and the structures of the respiratory system, the air sacs, and the tracheae. A whitish-yellow tissue, the fat body, is distributed around the organs. In insects, this tissue is comparable to the liver and fat of vertebrates. The respiratory system of insects consists of a network of tracheal sacs and breathing tubes, the tracheae, distributed in the hemocoel. The tracheal sacs are membranous and constitute an air reserve. They also reduce the density of the insect's body. The tracheae are chitinous tubes characterized by a translucent appearance. They form a complicated network throughout the body. In locusts, the air penetrates into the tracheal system by way of the spiracles that open at the surface of the integument. Then it goes into the tracheae that branch and rebranch into thinner tubes, the tracheoles, to penetrate the tissues where the oxygen arrives to cells by diffusion. The animal regulates its breathing by contraction of the abdomen as well as with the opening and closing of spiracles. The digestive system of the locust is formed of a complete digestive tract going from the mouth to the anus. It's divided into three regions: foregut, midgut, and hindgut. To observe these regions, the digestive tract must be removed from the animal. Remove the tracheal sacs and most of the tracheae surrounding the intestine. Take out also most of the fat body spread in the hemocoel. Carefully remove the tracheae around the gonads. Cut the digestive tract at the level of the esophagus and at the rectum. To remove the digestive tract, pinch the esophagus and pull carefully upward by sliding it under the gonads that are maintained in place. Place the digestive tract in the water. The foregut, or stomodium, includes the mouth, the pharynx, the esophagus, the dilated crop, and the proventriculus, or gizzard. The midgut, or mesenteron, has 6 blind-ended diverticuli, the digestive ceci. It's followed by a straight tube, the ventriculus, or stomach. Many small brownish Malpighian tubules are present at the point where the midgut joins the foregut. These tubules constitute the excretory system. The hindgut, or proctodium, includes a short ileum, the colon, and the rectum. The rectum is marked by 6 longitudinal ridges and bears the rectal papillae. In both sexes, the reproductive organs are located in the abdominal region. The gonads are 2 large masses positioned dorsally to the digestive tract. In the female, as well as in the male, the intestine must be taken out in order to observe the whole reproductive system. The female reproductive system is composed of 2 gonads, the ovaries, 2 oviducts, surmounted by 2 accessory glands, 1 spermatheca, and the vagina. A pair of ovaries occupies the anterior half of the abdomen. At sexual maturity, each ovary consists of a variable number of ovarian tubules or ovarioles, containing bright yellow ovocytes. The ovaries are ventrally connected with two large oviducts. The two oviducts join beneath the nerve cord to form the vagina. To observe the vagina, remove the ventral nerve cord. Then, the muscles of the ovipositor valvuli and finally, unroll the seminal duct. The seminal duct is moved laterally with fine forceps and fixed around a dissecting pin. At the upper end of the seminal duct is now visible the spermatheca, the receptacle used to store spermatozoa after mating. To observe the vagina, the tissues over it must be removed. The vagina ends between the bases of the ovipositor valvuli. The seminal duct opens on the dorsal side of the vaginal opening. The male reproductive system includes two gonads, the testicles, two vas deferens, two accessory glands, an ejaculatory region, and external genitalia. Testicles form one yellowish mass. Each testicle is formed of hundreds of fine tubules which join the vas deferens. To observe the beginning of the vas deferens located on the ventral side of the gonad, the testicular mass must be turned dorsoventrally and fixed with a dissecting pin. The beginning of the vas deferens is now visible. The vas deferens goes along the median part of the testicular mass and opens into the ejaculatory duct behind the accessory glands. Unroll one of the two accessory glands to observe their structure better. Each gland is formed of 15 coiled tubes with different aspects and lengths. The vas deferens and the accessory glands end at the ejaculatory duct, which extends into an ejaculatory sac bearing chitinous structures, the external genitalia. To observe them, remove the conjunctiva membrane at the base of the ejaculatory sac and fold down the subgenital plate. This procedure makes it possible to observe the external genitalia, the epiphallus, equipped with hooks, and the aedeagus, the intromission organ. The external genitalia maintain the abdominal edge of the male firmly hooked to that of the female during mating.